Hello, welcome to the 2021 Winter Field Day. My name is Clint Maddox. I'm a postdoc here at Oregon State University, and this short video is going to highlight some work we're doing on the carbon sequestration study. The first part of the video is going to highlight Dr. Claire Phillips from the United States Department of Agriculture, where she's going to explain the big premise of why we're doing a carbon sequestration study and the partnership that OSU and USDA is working on, in particular with this project. Following that, Dr. Renee Wang and I are going to highlight how we're actually doing this in the field on a bi-weekly basis, where we're going to be collecting carbon sequestration data on mowing, fertility, and irrigation studies at the Lewis Brown Horticulture Farm.
Hi, my name is Claire Phillips. I'm a postdoctoral soil scientist with the USDA Agricultural Research Service here in Corvallis. And I'll be telling you about a project we're doing on carbon sequestration. So the origin of this project was last fall when I was attending agronomy society meetings, and I started hearing, I attended a number of meetings along the theme of perennializing forage systems and row crop systems to try and increase soil carbon sequestration. So I started to think that maybe there is an untold story here in perennial grass systems in the Willamette Valley, both in our seed cropping systems and in our turfgrass systems, about their potential to sequester soil carbon. So last winter there was a grant opportunity, and I, together with my supervisor Kristen Tripp and with Alec, we put in a proposal that will start this October and run over the next 2 years, where we'll be looking at the carbon balance of a Pacific Northwest cool season turf system. And then the other piece of this is looking at the impacts on carbon balance of maintenance practices. So we'll look at mowing height, mowing frequency, irrigation, and nitrogen fertilization. So when I talk about the carbon balance of a system, I mean over a year, is the system, the soil and the grass, a net source of CO2 to the atmosphere or a net sink? And there's two elements that play into this. One is the biology, how much carbon is being taken up by the plants photosynthesizing and microbes respiring, and then there's the maintenance. So what are the emissions associated with mowing and fertilization? So to look at the biological part, we're going to be using flux chambers, and here's one of the flux chambers that just arrived. We'll have automated flux chambers as well that over the next 2 years will be measuring continuously over 2 years the CO2 uptake or release. So when we put a chamber on the turf grass, if the sun is shining, the CO2 will drop down, and that's an instantaneous measurement of the CO2 being taken up by plants through photosynthesis. If you cover up the chamber, make it dark in there, you'll see a CO2 trace that starts to climb, and that's CO2 being released by the plant as it's growing and putting on biomass, and also from soil microbes that are decaying the organic matter. So summing up those fluxes over the year gives us our balance and our estimate of how much carbon the system's sequestering or releasing. So we'll have two automated chambers, and then we'll also have, this is our survey chamber. We'll be working with the school IPM plots that have been in place for a few years, where there's differences in mowing height, mowing frequency, fertilization, and irrigation. And every 2 weeks we'll go out and at midday, when plants are at their peak photosynthesis, we'll compare how much CO2 they're taking up, and then we'll cover the chamber and see how much CO2 is being released at the same time by the soil. And that'll give us a sense of how low input maintenance practices affect the growth and the potential of the turf grass to sequester carbon. So we hope this information will be useful to the industry to help them explain their environmental impacts to a concerned public. The whole turf team is helping out with this project, and I think we're pretty excited to be pursuing a new research direction, and next year we should have some initial results to share with you all.
In this video, we're going to be showing you how we are collecting our carbon sequestration data. So we have constructed a light box with a lot of different basically light bulbs that provide the full spectrum of photosynthetic active radiation. So that allows the plant to undergo photosynthesis underneath that light canopy box. While we're collecting the carbon sequestration data, we are flooding it with light that is similar to the amount of light that we get per month. So we change the kind of bulbs that we put in there every month and where we turn them on and off. At the same time, we're collecting the volumetric water content percentage and the soil temperature. We have placed some basically PVC pipes into the soil, and on top of that PVC pipe, we place a dome that captures all of the carbon dioxide gas coming off of that particular area of turfgrass. And we're collecting this carbon sequestration data on 3 primary locations. So where we're doing different mowing frequencies and timings, where we're doing different irrigation frequencies, and we're doing different fertility regimes. So Dr. Rennie Wang and I are doing this and collecting these data every 2 weeks. And but this is basically telling us is the influence of those 3 cultural practices on how turfgrass uses carbon dioxide, how it undergoes photosynthesis. And so as it's capturing this carbon dioxide, the meter that we use to collect this data is recording the carbon dioxide concentration over that turfgrass. And as we look at a graph going from left and right, we see a little line that starts to build. And we see the number C, which stands for carbon dioxide. Here the number is currently about 336, and it's going to keep going down. What that means is the carbon concentration inside of the dome is decreasing because the plant is using that carbon dioxide to undergo photosynthesis. It's using that carbon to build turfgrass leaves, roots, shoots, etc. And then we also do the same thing in the dark to see how the soil microbes are affecting the carbon dioxide concentration over the turfgrass stand.